So welcome everyone to this month's version of the ReproNim monthly webinar. So the first 14, 15 months of the ReproNim webinar have really been fairly concentrating on ReproNim technologies. And so that's great. So we now have a lovely collection of lots of videos and questions and discussions about sort of the technologies from the technology-centric perspective. As we enter phase 2 of ReproNim's, hopefully phase 2 of ReproNim's support, we are kind of required to be a little more user-focused, user-centric, and really spend some of our time really being sure that the technologies that we're developing are being used in the outside world. So with that philosophy in mind, I'm going to try to pivot, famous word of pandemic, we're going to pivot the presentations for the next year or so, and we'll see how that goes, to a little bit more biology or application area first. So to try to talk about some papers, some topics, some biological topics that ourselves and our collaborators and others are working on, and try to then pair that with how the ReproNim technologies affect the biological question that's being asked. So we're going to try this for a while. Some of these will be just folks speaking. Others may be pairs of applications folks paired with ReproNim folks. And again, some will be people who are using ReproNim technologies. Others will be people not using ReproNim technologies, and the conversation can be around, okay, what's the barriers and differences in order to use ReproNim technologies for those kinds of problems going forward. So this is an experiment, and we welcome all of you aboard, and anyone who wants to volunteer. We're always happy to have folks give presentations that help demonstrate ReproNim technologies or the need for ReproNim technologies. So in this vein, and sort of in transition, I figured I would grab this first session and look at one of the works that we're working on together, sort of within the ReproNim team, and that's to look at IQ and typical development. Using the ReproNim technologies to create a re-executable version of a meta-analysis that is driven and motivated by the historical literature on IQ and brain structures. So while I guess nominally I'm the one assigned to be the speaker, there's lots of elements in this that's really a team effort. So I may surprise folks on the call and make them answer questions as we go along. The usual stuff is on the slide, and we thank you all for attending today. So while I wanted some biology in these presentations, I'm not actually a brain scientist, and so I don't have huge insight into how IQ and brain are deeply integrated together from a biological point of view. But what I can say is that alterations in brain structure and function that are associated with IQ have been implicated in a huge number of studies. IQ, like sex and handedness and age and other key demographic factors, are highly likely to exert relatively large influences on the brain structures, functions, connectivities, et cetera, that we observe. And those large influences are therefore an important source of variation that must be accounted for when we're trying to elucidate other more subtle influences of disease and disorder and maldevelopment types of things. Even asking the question of the literature, okay, what do we know about brain and IQ? And we want to start from typical development. What do we know about typical development and brain and IQ as sort of a starting point to trying to understand various other pathological conditions in the future? I hate PubMed. No, I didn't say that out loud, but asking these kind of practical questions of a resource like PubMed, our main ability to search the published literature, ends up being not at all very useful in the specific question of studies that look at brain and IQ in typical development. You can get lots of answers when you query it, but to really turn it into studies that look at quantitative measurements of IQ and the ones that look at typical are very troublesome. So again, you can wield that, but in the end, you have to kind of manually go through that and manually understand how to extract the publications that are relevant to that particular question. So one can do that, of course. But gee, wouldn't it be nice if ReproNim or other folks made it easier to query for more specific results from studies, so that the question of IQ and brain and typical development could be asked of the data as opposed to the abstracts and free text that things like PubMed searches. Maybe a job for ReproLake in the future. Anyway, so from that huge literature, our group, over the past few years has identified a handful of just sort of exemplary publications from the literature that look at the question of IQ and brain features of various sorts. So we've sort of isolated 3 exemplary papers and looked at sort of what their summaries are, and we're going to use those as a starting point to see if we can recreate from that literature review the findings in shared data using our ReproNim technologies. So there's 3 papers that we're sort of using as this jumping off place. The first of which is this one shown on the screen now, a meta-analysis from the published literature that looks at the associations between human brain volume and intelligence differences. How strong are they and what do they mean? So this is a meta-analysis of the existing literature. It looked at 148 healthy and sometimes clinically mixed sex samples. About 8,000 individuals were included in those 88 studies. And they document in this meta-analysis a significant positive association between brain volume and IQ. And that generalizes over age, children versus adult, and over the various IQ domains and over sex. And so to not belabor the details of this, but this is the 88 typical studies that they've looked at from the literature. Again, all showing, or many of them showing, a slight positive correlation between brain volume and IQ, and the meta-analysis here, sort of documents a significant IQ brain volume relationship in typical subjects, and these are ordered from the long past back since 1991 to the relative time of this publication back around 2012. So again, a lot of studies, some of them are all over the place, but in general, again, pretty consistent observation of a significant positive correlation. And it's kind of interesting that in this case, all the studies since sort of the middle of 2010, all were kind of significantly positive, despite so as sort of technologies advanced and things like that. Whereas before that, it was bigger error bars and often hitting the 0 effect more often. So anyway, but overall, the literature is very consistent with a significant positive effect, brain volume, IQ. Great, one thing we should be able to check. The second exemplary publication we're working with is investigating the relationship between striatal volume and IQ. This paper here, ultimately, they observed that correlations between a particular IQ measure, the WASI IQ, and the left striatum. And for those of you who don't remember, the striatum is a composite structure composed of the caudate nucleus and the putamen. So the correlations with IQ and striatum in the left hemisphere were significant. And when analyzed separately for the male and female, the positive correlations were significant for the male only. So again, nice little simple question to be able to ask of our current data and our current methods. Can we replicate that particular finding? And a figure from the paper showing this moderate correlation of left striatum and the WASI IQ. And the third example looks at negative association between corpus callosum mid-sagittal area and IQ in a representative sample of healthy children and adolescents. This comes from the NIH pediatric database back from the mid to late 2000s. 500 or so subjects measured at 5 or 6 different sites. It's really the first of the sort of big data initiatives funded by the NIH to try to collect large scale data sets on development. So this is an exemplary publication from that, showing a significant negative correlation between the total corpus callosum mid-sagittal area and IQ, and it tended to be the negative correlation was in the males but not the females. So again, nice simple compact observation to attempt to replicate. And just again, representative figure from here, reminding us that the corpus callosum area and brain volume, are associated, as we, and then the IQ is related to, in a negative way, to the area and the IQ. So from those 3 publications, the specific set of hypotheses that we can generate from all the observations embedded in there, are enumerated here, and the source, code that we're using. So we have some that are related to the total brain volume, positively correlating with IQ in both sexes across the complete age range. Others have to do with the left striatum volume, positively correlating with IQ in both gender, both sexes, but maybe in the males only and not in the females. And then we have these corpus callosum measurements that suggest that there's a negative correlation with IQ, across the group, and that these are different, in the younger group and different by gender. So those are sort of the 9 hypotheses, that come out of those 3 publications. So we're trying to do everything, by the book, sort of in the ReproNim-ish way, and that, of course, includes, from our hypotheses, we should do a pre-registration, so that we have, undertaken, some of the exercise to pre-register these hypotheses. Yeah. And this is the same, content, but just a little bit more geometrically aligned. So each of these, hypotheses have to do with a particular brain region and its relationship to IQ, a correlation direction, a age subset, and a sex subset, from the sample of data of typical developing data. So these are the 9 hypotheses that we're going to try to do, in contrast to the existing literature, which is, not particularly reproducible, not explicitly reproducible. We're going to try to test, these 9 hypotheses in a way that is documentably and verifiably, sort of reproducible, going forward. So these are the 9 hypotheses. So just a quick refresher on the ReproPub, the Re-executable Publication concept. So we're trying to really, use ReproNim technologies to move beyond our usual, publications, which tend to be words, in a PDF, file, but make sure that those lovely words that are necessary for the human interpretation of what we're doing needs to be accompanied by an exact specification of the data that went into that particular publication, the workflow specification, the environment specification, and the complete results of the processing and of the statistics specifications, so that one can re-execute it and verify that re-execution is successful via these executable elements. And the idea is that a completely re-executable publication requires the complete description, the complete provenance of the data, workflow, execution environment, and results. So that's the concept that we're trying to work from, and trying to put these together into a more practical, real world example here. So we have these structural correlates of IQ in typically developing children hypotheses. We'd like to explore these observations and also look at some of the known methodological dependencies that we know about. Different data matters, different software matters, different operating systems can matter. So we want to be able to do this observation in a way that lets us explore those dependencies in a more systematic fashion. So the tasks to accomplish this kind of re-executable publication means we need to discover what data is relevant to our particular question. So we need structural MRI, we need typically developing subjects, we need cases that have IQ and age and sex. We need to pre-register when possible, which we've done in this case. We need to be able to annotate the sources of data. In this case, we'll be using multiple sources. We need to get the IQs and age and sexes, which may be coded differently, all into a sort of a standard representation so that we can interact with that data more seamlessly. We need our workflow to be specified and the workflow to be operationalized into a container of some sort. What tools do we need to use to accomplish what we're after? What operating system do we want to use? Where are we going to execute this, locally, local high performance, cloud, et cetera. After we've run that, we have a bunch of results. We need those represented in a proper annotation, form. And then from that, pile of results, we need to be able to query that to extract the specific subsets that we need and perform statistics again in a controlled way where the provenance of the statistics and the results, are captured. So this is a project that's been ongoing, for quite a while. So today, we would go to ReproLake to try to ask the question about, okay, what do we know about, data that has, IQ and things like that. But back at the time, there was this lovely resource, Satra, Roberto Toro, and others have been working on called Metasearch. So this is, what we used at the time, where we were able to specify, okay, we want typicals. Okay, well, let's just pick the controls, from the subset of data that they were sampling over. Well, we want people who have, IQ measured. Okay, let's sample, select, the subjects who, have IQ. And so it turns out that of the data that Metasearch knew about back at the time, the only two data sources that, had data for controls and data that included IQ turns out to be the Abide and the ADHD 200 data sets. So fine. Now this answer, may change over time, and that'll be interesting. But at the time, that gave us two data sources that helped, sufficient, to make our observation, and provided sort of 935 pointers to subjects that would fit, this criteria of having imaging, having, being a control, and having IQ and sex, annotated. So fine, so that's how we, in this case, discovered, Abide and ADHD 200. But again, that might be done differently, going forward. So given the need that we've identified that Abide and ADHD 200 are the data sources that we're after here, we were fortunate to realize that one of our local resources, DataLad, already knows about these data sources. So DataLad gives convenient ways to access the Abide data, the ADHD 200 data. Now it's not been annotated at this point in time, but at least it had been represented in BIDS format and is easily downloadable. So since DataLad had already picked up these data sources, the tasks for us in this case then, or David Keeter, who is now here, that was great, is to be able to add the annotation for each subject, the sex, the IQ, the diagnosis, from the source data, and again sort of resolving sort of inter-source and inter-site variation in these behavioral codings, and to provide the NIDM annotation for the imaging. And since this data was BIDS, that a lot of the BIDS representation image data representation, was relatively straightforward. So again, big mess and trouble with the variation and how different data sources, annotate, express, these various behavioral information, and that has to be solved. And thanks to hard work by Dave Keeter, that was accomplished, so that we now have the Abide, the ADHD 200, and other data sets that have uniform coding for sex and IQ and diagnosis, and these other important variables for this particular study. I'll come back to this slide once or twice. This is kind of the overall sort of data model, the representation of the neuroimaging data model of various different elements that we will be talking about. So ultimately, we have subjects. Those subjects have demographic assessments, so those need to be codified properly. Subjects participate in imaging sessions. Those imaging sessions have multiple images associated with them. Those images need all their provenance and metadata associated with it, documented and linked together. So in the sort of linked data concept, we should be able to go to an image, know what session it came from, know what subject that came from, and discover what behavioral or demographic information is available. Ultimately, later, we'll talk about running analyses on particular images. So each analysis has a provenance and metadata track that's associated with its application to a particular data source, and generates resultant information that will codify as well. That'll come up a little bit in the future. I'm going to throw Dave Keeter right on the spot for just a second and say if there's anything else about that annotation that he thinks would be relevant to point out here.
I just wanted to say it wasn't that hard.
Okay.
We built some good tools.
Now, I guess there's a disclosure that those tools are his, but okay, great. Good, thank you. And if I missed anything, you'll update it in the notes later, so thanks. Okay, so fine, we've gone through the process. DataLad delivers the data to us. The NIDM representation of that data, codify properly the annotations of the demographics, the imaging metadata, and now we're ready to do some processing. So from the hypotheses, we have only a handful of target results that we're after from the image analysis. We need something about total brain volume. We need something about the left striatum volume, and we need something about the corpus callosum area. So the tools again, there are many tools that one could use for that. In this case, we have access to FSL, the FIMRIB software library, FreeSurfer, and ANTS are all tools that were readily at our disposal that generate results relevant to these particular questions. We already know that different tools sometimes cause differences, so it's nice to be able to use sort of 3 different tools so we can compare and contrast the effect of different tools, in addition to some of these tools do and don't make all of these measurements. So it's just a collection of tools we had access to create this imaging derived volumetric derived data set. And then a miracle occurs. Somehow the workflows are assembled into in this case sort of NIPIPE workflows. We had done a paper a while ago with the horrible simple one title, which is really just an FSL workflow that takes imaging data and generates the subcortical segmentations. So for this task, we need the FSL workflow. We also need a MindBoggle workflow, and MindBoggle, Arno Klein and his colleagues, is already sort of pre-combined, FreeSurfer and ANTS into a nice joint effort, and expresses the volumetric measures in terms of the FreeSurfer segmentation parcellation system. So I'm going to call on Satya right now if there's anything he needs to add about how these workflows were created, if he wants to or not. For the record,
they're all containerized workflows, so it's easy to kind of pull and run. I think the one difference we made was we combined the simple one workflow and the MindBoggle workflow into a common container.
Okay, so that was one of my questions, and thank you for resolving that. Is it, in principle, one could run 2 containers, but in this case, you could also combine them into 1 container, depending on your needs and what's convenient for your particular problem. So fine, we have these different workflows. They're enabled in NIPIPE, I presume, and are containerized and accessible to us. So really, so you're fine, you run that, and now we have 900 more subjects that have all of these results that are generated. So we now work over on this far right side of this, where each of our different software tools generate a set of results. Each of those results have a annotation, have a meta analysis that or metadata that is associated with them, and so those are annotated. And again, this relatively simple in the sense that it has to only be done once. Someone has to teach software using PyNIDM how to encode FreeSurfer results, how to encode FSL results, how to encode ANTS results. But once you've done that, it's relatively straightforward to apply those annotations to any future runs of those software. So now each of those results can be codified in this sort of standardized fashion. So we can then more easily find comparable measures across the different tools. So a volume of a total brain from FreeSurfer versus the volume of total brain from FSL versus the volume of total brain from ANTS can now be more easily extracted as a query on, okay, give me the total brain volume, to which will return, okay, there are 3 different tools that do that. And of those tools, here are the measures from those tools that relate to that particular query. So this is really where all that tying up of these multiple tools comes together, so that one can sort of dynamically discover and then apply different applications to the results of the different tools together. So fine, lots of data codified now. So we have lots of NIDM. We have NIDM for the subjects. We have NIDM for the imaging. We have NIDM for all the results. So lots of NIDM. So we use the query functionality from PyNIDM in order to extract specific subsets of that meet the particular needs of the different questions that we're asking. So from that, one can extract the sort of a data frame that's necessary. And then from that, one can build the various statistical analyses that we need to execute the queries, the statistical analyses that are necessary to confirm or deny the hypotheses that we're starting with. The analyses are in GitHub and are therefore shareable and version controlled in that particular way. I'm going to ask JB if there's anything else he wants to say at this sort of high level about the statistical approach to this.
Nope, I think you said it well. Thank you.
Okay. So fine, we have data, gobs of data. We extract that with the Python IDM query, and we ship that into a IPython notebook, Jupyter notebook, and run the various queries. So I guess I'm going to jump right to the result, and then we'll work backwards about a little bit of the details of the result. So again, we had these are 9 hypotheses, and we can now ask if in our new data, do we confirm or deny those particular hypotheses? So just looking at this first row, we had the hypothesis that total brain volume should correlate positively with IQ across all of our ages in both of our sexes. We see that if we use FreeSurfer and ask that question, we confirm that we have a very significant correlation between IQ and brain volume, and we're covariant for sight in this case. We get the same answer with FSL, we get the same answer with ANTS. So right here on this particular hypothesis, we're all looking pretty good and all pretty happy. The new data, new analysis approaches confirm that meta-analysis from the literature, and it doesn't matter which tool of these that we're using, and the significance is pretty robust. Great. The left striatum volume, FreeSurfer, FSL, ANTS confirm this positive correlation between left striatum and IQ in our subset of subjects that are less than 20. And again, which tool we use doesn't matter, and that's confirmed. We confirm that in our male subjects, we still see this positive left striatum IQ correlation. Again, doesn't matter which tool, but we nicely confirm that a priori hypotheses. MAC3, okay, now it becomes interesting. So the original publication that we were looking at claimed there to not be a significant relationship between IQ and left striatal volume, in the female subgroup. So we didn't confirm that in the sense that we do see a significant IQ correlation with left striatal volume, across all 3 tools. So this begins to be interesting, and kind of the types of things that we want to understand is what hypotheses, what claims from the prior literature confirm and which ones don't. Now again, we have to remember this is different data and different tools. So this is just one example of the attempt to generalize that particular finding. And so where they didn't find a correlation with IQ in our particular data, we do see a correlation with IQ. And it should be noted that we're looking at about 130-ish subjects in our data set, and the original paper was probably similar size subset of data as well. So it's not necessarily the number of data points in this case, but again, we're seeing a different answer, different tools, different data. So this is exactly the kind of fodder that we need to be able to dig more deeply into. Is it the tools compared to that original publication? Is it the data? What is causing this type of difference? And then relative to these corpus callosum observations, the given hypotheses, we're expecting to see a negative correlation with corpus callosum area in a number of these groups. We did not confirm that. We're seeing no correlation in these particular groups. Again, we're covariating both for sight and total brain volume. In this case, we are using substantially larger data sets in these first 2, whereas we have access to 535 subjects in this cell, where the original paper was 198. So we aren't confirming that negative correlation. Now again, different tools, and truth be told, we're looking at corpus callosum volume, not corpus callosum area. So fine, that might be part of the difference. But again, we have to use this kind of discrepancy, not to just wish it away, oh, different methods, but to be able to dig more deeply, specifically into, okay, what is causing that? Is it really a difference between area and volume? Is it really a difference between the software? Is it really a difference between the data sets that are going into this? We confirmed 2 of the corpus callosum findings. There was a finding that said that the subset of subjects that are greater than 12 should have a non-significant corpus callosum area, and we confirmed that. and the subjects less than 12, females should have a non-significant correlation, and we see that as well. Now I know JB had some questions about when we're looking for no significant correlation and how we prove non-significant correlation. I don't know that we'll discuss that now, but we could come back to that topic, that when the hypothesis is to not see a significant correlation, what the sort of right way to test that is. So the moral of the story, though, is that amongst these sort of corpus callosum area measurements, there is a good bit of variability in between what we're seeing and what the hypothesized effects ought to have been. But again, we know there are differences in tools. We know there are differences in the measure. We know there are differences in data sources. That's not so much the problem. We just need to be able to work with that in order to understand the sources of these differences. Given that we're having some issues, in these corpus callosum area types of measures, we also, sort of added some post hoc, questions. That particular paper, had suggested that corpus callosum area and total brain volume, should correlate with each other, which we do confirm. That paper also suggested that the corpus callosum area and age, should, correlate with each other, which we actually don't, see. So again, there's ample evidence that there's some differences, in these two approaches, and again, that's fine. We just need to understand them, sort of appropriately and systematically. So let's dig in and look at some actual pictures of some of these observations. So this is that so first, well, the three McDonald hypotheses, one of which is that in boys and girls who are less than 20, we should see a correlation between the full-scale IQ and the left striatum. So this is our so we're using FSL, free surfer, just showing different examples of all these ones that are significantly correlated, and recreate expected positive correlation, and we observe positive correlation in these different subsets of subjects. Great. Examples of these latter ones that we don't see in the full-scale IQ and corpus callosum volume measures. We were expecting a negative correlation, and we're observing a non-significant one, both in the under 20 both genders, the under 12 both genders. We're expecting negative correlations and not seeing it. The greater than 12 both genders expected non-significance, and we confirmed the non-significance. The less than 12 males expected a negative correlation that we didn't see, and the less than 12 females expected non-significance, and we observed that non-significance. And then just to look at the more basic question, again, we are seeing substantial corpus callosum volume correlating with total brain volume, but we're not seeing corpus callosum volume correlating significantly with age, and we're not seeing total brain volume in this group correlating with age. So again, important things to understand, and the really the jumping off place to again a systematic understanding of why one result is different than another. So this example sort of fits the ReproPub concept, in that there is a place where you can go to see all of our pre-registered hypotheses. There is a set of containers that you can go to that will run these workflows. There are annotated raw data sets and processing results at particular target locations. There is the statistical analysis version controlled and present and linkable to, that can regenerate these results from the source data. So in principle, any reader of this article should be able to get to and recreate the analysis, get to the data, regenerate the statistics, all from the content of the article itself. So just to sort of wrap up, again, conceptually, the comparing and contrasting of a traditional publication with a ReproPub. Again, all of our hypotheses were generated from traditional publications, which had lovely plots, had lovely descriptions of their methods, had lovely statistics, described the effect sizes and correlations and all that kind of stuff. That's fine. So humans could read that, but the ReproNim variant of that really wants to make sure that every element of that is itself, re-executable. So every point, on the ReproNim, re-executable publication, knows, what subject that came from, knows what MRI scan that came from, knows where that scan, is living, knows what workflow, generated this volume measurement, knows, has pointers to the container, that generates that, knows where this measurement, fits in the constellation of other measurements. So given a new, so any individual, given the ability to access that data, could regenerate that data point. And given new data, one can generate data points of the exact same method, or if one wanted to explore and change that method, one could do that as well. So each of these points, therefore, are both re-executable, and that anyone could re-execute this point, but it's also generalizable, since anyone could change the data or change the analysis in structured ways and understand how that affects that analysis. Similarly, the statistic is regeneratable and extensible to new data, and version controlled, and executable by any reader of the observation, and extendable to new data. So in contrast, so when we if someone redid our paper and found differences, that gives them a way to therefore better probe the differences. Whereas right now with the non-re-executable traditional publication, there's still a lot of guessing about the types of things that could cause differences, but not good way to understand which of those things did cause these differences. Now in this case, phase 2 of this paper actually can go back to these. I mean, these are publicly available data from the pediatric database. The tools that were perhaps used in a couple of those publications were probably Civit or something that we could get access to. So again, the opportunity to go back and really understand why one version of this replicates, and why one attempt to replicate this doesn't, and better to be able to understand sort of exactly where the data and exactly where the tools are diverging in that understanding. So that's about all of my planned presentation. I will end with some new motivational material I'm trying to use. Reproducibility, just do it or else. If you don't do it, you won't get funding, and that's reminding us all that there's new policies coming forward, that are much more strong about data sharing and data management. Also, many of your NIH grants won't get their subsequent year money if you're sharing and acquisitions are not staying on their target. Many journals now are really beginning to enforce their data and software sharing policies. So again, if you're not careful, you won't get published. And if you don't publish and get money, you won't get tenure. Just kidding, no one gets tenure anymore anyway. So that's just the but if you don't do reproducible research, John Oliver will laugh at you. And we're not going to play the episode now, but there's a link in the slides to a lovely John Oliver episode on the irreproducibility of science and how amusing, quote unquote, that can be. And worst of all, we really need Jarek to remind us that to really take all of these things seriously and adopt reproducibility by design. So that's sort of the end of this sort of application talk, trying to get through our concept of doing an interesting re-executable publication, in this case attempting to and occasionally succeeding and occasionally not succeeding at replicating some prior literature findings using publicly available data and sort of standard current workflows and containerized environments with everything marked up and annotated, so that the complete provenance and the complete generalizability and re-executability of the work is documented. Lots of folks are part of that. I've called on some of them to give their 2 cents as we've gone through. And there's all sorts of information about ReproNim that you probably already know how to find. And while we'll take questions next, I did want to advertise the next in our series of sort of application and ReproNim pairings on next month's webinar will be Anna Manelis from Pittsburgh and Jarek looking at the interplay of depression and obesity using ReproNim technologies and imaging. So I'm going to stop there and invite the audience to carefully unmute themselves, ask questions, and if the ReproNim team wants to add anything to that, they're of course welcome to as well.
Okay, yeah, I've got two probably very simple, silly questions. But the first one is I wanted to ask more about the slide you had, where they said where you found the data sets that had everything you needed to test the hypotheses. Was the software you used for this called ReproLake or?
So the software the example I showed was something called Metasearch. I guess I could go back to my sharing. So this is the example here comes from a tool called Metasearch that sort of precedes, sort of the ReproLake, types of concepts, but does sort of a very ReproLake-ish thing. And Satra and Roberto and other friendly folks, that we know, developed this particular interface. But it operates on a handful of known data sources and gives you a lovely intuitive interface for how you can search and do subsetting of those data based on various elements. So this is Metasearch. I threw out the concept of I introduced ReproLake here because this is really what ReproLake is trying to extend and be able to give that same sort of functionality, and someday with this Qt a front end interface to be able to access the ReproLake content.
Thanks, that's good. And the other question I had was, you mentioned that for each point on the graph you created, you know the things like the subject, the acquisition details, the pipelines, etc. So in data sets that you have to register to use or have different privacy settings, are there any privacy concerns with publicly showing the details for each of these points? Or is it fine to feature these details in, say, a website or a GitHub or something like that?
So that's interesting and important distinction to make. So in the general world where some data is protected, we do have to be a little bit careful about some elements of this. So even though we know who the subject is, if an observer can get to that subject will depend on their authorization. So again, if this were ABCD data or other things from the NIMH data archive, no, unless you would know something about it, but you'd not be able to resolve that unless you were duly authorized to get there. The workflow that was used most often will be publicly accessible and not behind some firewall. So again, if you can actually re-execute it or not, in this case, doesn't depend on your access to the container, since that should be accessible, but it depends on your access, your personal access to that data, which maybe you get access to or maybe you don't. And so again, that depends on access. If you really want to get it, then you better figure out how to get it. If you can't get it, well, then you can at least try to find similar data and ask if you can get that.
And if you have access to the data, is it still always fine to publish these things publicly? Like, say you can get access to the data, is it still fine to put what subject is and what the scan is?
Yeah, that one is variable as well. And again, using, for example, ABCD as a good example, a good example of a problem set case. Their data use agreement may actually not make it permissible for you to share the derived results. So if this were a free surfer, you might not be permitted to, make the free surfer available. You need, in the ReproNim way, you want to make sure that is someplace where that an authorized person could get to it, but that you can't make that, generally publicly accessible. So NDA and ABCD, for example, use I'm forgetting the term, but there's ways that if you have NDA access, there's ways for you to return derived results back to NDA collections, so that one can do this, except that the raw data is living behind the protection of the NDA, and the derived result you generate may be living back in behind the protections of the NDA, so that again, an authorized person could follow that, but an unauthorized person could not follow that exactly, and therefore has to deal with that.
Okay, thanks a ton.
Sure. And if anyone else from, Peter.
Just a couple additional comments. The the ability to, like in Repro Lake, what we want to store are, metadata, representations of the metadata, but not the actual, data set that you might need for analysis in the case of these sort of protected data sets. And the granularity at which we'll be able to essentially share the metadata in Repro Lake is, dependent on the access restrictions from the original data sets. But, that being said, in some cases, if we can store some, a little bit of data along with the metadata, that makes the searches in Repro Lake more powerful. Searches like, find me subjects of a particular age range with a particular, IQ measure in this example. The IQ measures, in order for that query to work, the IQ measures, need to be there or be accessible to perform that query right now. So in that case, you've got some metadata mixed with some actual data, the IQ measures in these NIDM files that then, facilitate the queries. But, getting a hold of the imaging data from there, in the NIDM files, of course, the imaging data isn't there, it's linked out. So, in that sense, you don't have all the data in these metadata files that you would need to sort of do the analysis, assuming that the imaging files are protected because of, access restrictions. So we can, do a variety of different makeups of the data in these NIDM files from just, solely metadata. This study had, 50 participants. Here's the age ranges of those participants. Here's the breakdown of, genders and so forth, to a little bit of data mixed with metadata, to then, I guess, a full, NIDM data set that you have access to all the images plus all the assessments. So I think, our approach will be not one size fits all, but dependent on the, and some of our, discussions of the future of the, sort of interface for Repro Lake would support those sorts of things where, you might be able to do a query and because of the protected nature of the data sets, of some of the data sets underneath, in some cases, you may just get some summary information, and then you would have to apply to each of those data sources to get access. And then from there, maybe you could, go back to Repro Lake and then do the query again and have richer information because you have access, that sort of thing.
Great, thank you for that. Other questions from listener land?
Hi.
Hi.
This is Dorian. This is a question somewhat related to what you were talking just now about data sharing. The data that is shared, I want to have a feeling, what's the feeling of this community? Because this is a question that comes up also for us. I'm in the Huntington disease community here, and we share data with others. Are data, publicly available data or data that are shared now, regularly defaced? And would the software that you used, at least in this presentation, be able to handle and work with defaced or blurred face data?
So the prevalence of defacing is a kind of variable thing. Again, depending on the IRB and the data sharing that goes into that. Some IRBs, didn't care about defacing and you can get away with that. Other IRBs, believe that face, and the MR reconstructions you can do from face, becomes identifiable and therefore requires defacing. So it is a bit variable in general, to the extent, the broad cases of very openly shared data are defaced. I think most of the Indy and Abide data set has been defaced. I see Satra has appeared, so I'm going to let him see if there's additional subtleties to that he wants to add. And most of the software we're using in this case, handles defaced data, but that is an important question that many tools can care about that. So it's important to keep into account that.
I'm just going to add that, in fact, there is a deface detector that's being developed, one that checks to see if somebody has posted data publicly that is not defaced, that is defaced or not. The Open Neuro Archive is using this to kind of clean up their data sets through that process and going back to individuals. Because in some cases, a person may have completely consented to share the data. So it has to be kind of taken on a case-by-case basis as to how the data got out into the public domain. The other piece to this is quite an interesting question in terms of software. There's kind of a dichotomy going on right now as people are building software on open data sets, which are mostly defaced. They may not work as robustly on other data sets that are coming out in clinical settings that are not defaced. So this is something that is important for the community to kind of keep in mind as we develop software on open data sets, that we also have to test them in settings where some of those processing methods have not been applied. For what Dave presented today, most of the software is used in that stream would work equally well for defaced and non-defaced data.
So I'm very curious to know more about that deface detector. So maybe I can shoot you an email later or something like that.
Great. Oops, I see something in the chat there.
And just to like, I know that
Thanks,
Per
Kendra.
has developed actually a little tool to help you deface. So because there's a number of defacers, there's a little ReproNim tool that I will put the link to that helps both in terms of the defacing and also in terms of the metadata that are potentially re-identifiable. I mean, problematic as well in the JSON file. So there's a little tool that may help in that respect.
I'm going to direct Kendra's question to JB about curious to hear a discussion on confirming past null results.
Yes, so it's all about power. It's all about like, defining what is your what is the thing that you want to reject. So you never be able to say, okay, if I had a null result, it means that my actual effects is within a sort of interval, right? And so you really want the power to make sure that if it was outside of that interval, you would detect it. So that's so it's a question of how do you set that or like how do you estimate that interval in which if the effect size if you're not outside of that interval, you can say, hey, this is a null hypothesis is visible. So it's really a measure of, it's a matter of what is the delta that you are allowing yourself to have for not rejecting the null. And people do that in clinical trials as well. I mean, like, they in clinical trials, you actually rarely reject the null. You reject something that is, if that is not beyond that delta of effect, then I will assume that there's no effect, or there's not a significant effect, but a biologically significant effect, which is a different thing from the statistically different effect.
So in this case, did you do a power analysis to show that where you got null results, you can be fairly confident that they are actually
I don't remember clearly when it's been a while. I think we had a lot of power because we had many more subjects than we had in the original sort of like in most of the hypotheses anyways. So in that case, I think we were kind of confident that that the non-significant was actually saying something. But I can't. I'm sorry, I can't remember the details. I should go back to it.
I don't remember that we did do that, and I think that's an important thing that we should do prior to publishing this, is to consider that more completely. Thank you for the question.
Can I bring up one quick thing on that note? I think part of this is if you looked at the effect sizes, which were, I think, in your second column of that table, those are still very small effect sizes. And that might be something also worth kind of considering, that even if it's significant, what is the impact of that relation? And I think the non-significant significant question should also be coupled with an evaluation of what's the meaning of the relation.
Yeah, and that's what like clinical trial people and others like call epidemiologists, like they call it biological significance. And that assessing the biological significance is what you're referring to, Sarah. And yes, and you're right. And again, you could have like a statistical significance always if you had enough data. Because there's no biological relationship that would be exactly 0 to the limit, right? So that's why assessing that biological significance is critical.
Okay, we're pretty close to the end of our hour. If there was a burning question, we could happily take it. Otherwise, I thank everyone for joining us. Hope to see you all next month. And you know how to find us if there's things that are less than burning that you still want to contact us about. So thank you all, and we'll see you again on the other side. Stay safe out there in these hopefully near end of pandemic times.